And then for the final talk before we come to questions, I'd like to turn to Nick Harvey from the University of Southampton, also a member of the Imaging Working Group for Biobank. He'll tell us about UK Biobank arthritis data and some new and exciting collaborative programs developing. Nick.
Well, thank you very much. Well, it's a great pleasure to tell you about the musculoskeletal aspects in UK Biobank. And I guess we've heard a lot about the cardiometabolic and brain aspects, so a slight change of direction. And I thought over the next 10 minutes, I'll just introduce the burden of disease which results from chronic musculoskeletal conditions, and say a little bit about our current approaches to risk assessment and how the gaps in that can be addressed through work in UK Biobank, and think a little bit about the DXA protocol and what it gives us and the image-derived phenotypes, and the work that's recently been funded by the Wellcome Trust to derive new phenotypes from the DXA scans. And I thought I would set this in the context of the emerging issues of multimorbidity, which is certainly very topical in our ageing population. So we know that musculoskeletal diseases, for example, back pain, osteoporosis, osteoarthritis, place an immense burden of disability globally, and so through the Global Burden of Disease Initiative, it's apparent that about 3% of GNP equivalent is accounted by the disability, the years lived with disability consequent to these conditions. And osteoporosis through fractures and osteoarthritis through joint replacement cost about £8 or £9 billion per year to the UK, and are really very common. So 1 in 2 women, 1 in 5 men at the age of 50 will have a fracture over their remaining lifetime, and over the age of 65, probably about half of us will get some manifestation of osteoarthritis. And so these are conditions which are really very common. And it's not just about those obvious events, but about the 20% reduction in relative survival following fractures that means that these conditions have an impact far beyond the obvious immediate effects. And this burden of disease is set in the context of an aging population in which increasing numbers of individuals have more than one common chronic non-communicable disease. And this issue of multimorbidity, addressed recently by NICE, is an area that UK Biobank is uniquely well set up to explore. And so if you think about the way that we do primary prevention for osteoporotic fracture, and we assess risk using the FRAX calculator, as you can see here online, gives us a 10-year probability of fracture feeding into our national guidelines accredited by NICE. But we know that doesn't identify everybody who goes on to have a fracture. And we know that there are ways in which we could make this better. We've done some of those already to modify the FRAX score. But UK Biobank gives us a fantastic opportunity to explore ways of risk assessment, as just an example, but linking the bone to other aspects, such as other body compartments, other diseases, other factors such as environmental and so forth, to generate novel markers of risk from the image-derived phenotypes, and to bring together measures from different organ systems, from the imaging study, for example, together with the mechanistic data, the genetics, the biomarkers that have been collected at baseline. And just to say very quickly about the DXA protocol itself, this is based on the LUNA IDEXA instrument you can see here, which is a state-of-the-art densitometer. And we have a whole body scan that gives you body composition as well as bone, and also some regional breakdowns. You can get android-gynoid ratios and so forth. A scan of the hips gives us bone indices and an image of that region. We get a DICOM image of the knees, and then of the lumbar spine, an image and bone indices. And then a lateral view of the thoracic and lumbar spine that can be used for vertebral fracture assessment. So I think one of the added value things of UK Biobank, and this coming back to Rory's point earlier about the sort of imagination of the users, One of the groups of scientists who've been most interested in the DXA data have actually been the computer scientists, the engineers around the images that come out. So, of course, we have the numerical data relating to bone and body composition, which are immediately available. They don't require any post-processing. They go up onto Showcase. They're there for use. But we also have these rather beautiful high-resolution images generated by the DXA, which can be used as you would any other radiograph, but also lend themselves to novel techniques. For example, shape analysis, automated methods for detecting vertebral fractures. And those images and their generation is the basis of the Wellcome Trust grant, which is very, very grateful. This was funded to the tune of £1.6 million by the Wellcome Trust through a collaborative award. And this is a study, AUGMENT, led by Professor John Tobias, who's Prof of Rheumatology in Bristol. And this brings together epidemiologists, clinicians, and engineers, computer scientists, and basic scientists across the centres you see there, Bristol, Southampton, Manchester, Aberdeen, and Cardiff, to try to derive ultimately derive better ways of stratifying the risk of musculoskeletal events. And the idea is that we develop automated platforms, and indeed these are pretty much sorted now, to generate these images generate IDPs, so image-derived phenotypes from these images in all 100,000 as they go through. And the sorts of things we will derive is, say, an active shape model, which gives you modes of variation in the proximal femur. And we know that from smaller scale studies, there's evidence that the shape of the femur is related to future risk of fracture and arthroplasty. We can look at signals for osteoarthritis, for example, in the knee and the spine, and identify vertebral fractures automatically. And using these as exposures, we can then link to incident outcomes such as fracture and arthroplasty. But we can also link to genotype and to the biomarkers to get an idea of the things that might determine these outcomes. And then look at further sort of causal type studies through Mendelian randomization and link to laboratory investigations to further characterize potential targets for therapy. And so the outcome of this, we hope, will be that we can inform better approaches to risk stratification, but also be able to identify potential new mechanisms and potential targets for intervention in future studies. And importantly, this will generate a library of new image-derived phenotypes which are fed back into the UK Biobank resource and are therefore there for future researchers to use in coming years. So I've asked this question, what's different about UK Biobank? Well, I think we've heard really throughout the day what's different about UK Biobank. And in terms of DXA, it's very much the combination of the numerical data and the images. And the images have added a huge new dimension to what's possible here. But of course, UK Biobank is unique in its size and depth and breadth of phenotypic information. And in the context of the things I'm interested in musculoskeletal research, it gives us this unique opportunity as we go forward to investigate musculoskeletal relationships in the context of other organ systems and other morbidities. And just to illustrate that, this just summarizes a few of the studies that are ongoing at the moment that I know about, and there'll be many others that aren't on here. But as we move from the baseline data of the genes and the myriad of data collected at baseline through the imaging enhancement that will eventually be of 100,000, and then to the incident outcomes through linkage, there are fantastic opportunities here and a wide range of things one can look at, for example, food environment in relation to musculoskeletal disease, got biochemistry, genetics, body composition, linkage to brain. A study that we're just starting, funded by BHF, relating bone to heart, with Stefan Peterson leading, and together with, of course, the Wellcome Trust funded work. So to summarise, UK Biobank gives us this unique opportunity through its breadth, depth, vast numbers to really address our questions of choice, not just in the context of a single organ system, as we would have been able to do previously, but in the context of pretty much everything you can think of wanting to look at, and phenotyped in exquisite detail and with state-of-the-art methods. And this is really highly relevant in our ageing society with increasing burden of multimorbidity. And it gives us the opportunity to bring together the observational epidemiology, the understanding of mechanism, to inform novel interventions. And that's not just therapeutic interventions, but ways of better assessing risk and of identifying those who need treatment. And I hope that the end result of all of this over years to come will be that we refine our ways of assessing risk and treating to reduce the burden of these chronic conditions. And so I'll just finish by thanking very much indeed the whole UK Biobank team and colleagues on the imaging group and the various other committees. Of course, the funders and then finally the UK Biobank participants, without whom none of this would be possible. Thank you.